Bismillah ar-Rahman ar-Rahim. Viral representation under supervision of Professor Dr. Sahar Abdulrahman, Head of Virology Department, Faculty of Veterinary Medicine, Mansour University. معكم الطالب نور مصطفى جابر. At first, what is a virus? Virus is an obligatory intracellular agent that needs susceptible living host cell to replicate its nucleic material and increase in number. So, virus is a non-living organism outside the cell. When it enters its susceptible living host cell, it is converted from dormant microorganism into vigorous entity. A virus is an infectious microbe consisting of segments of nucleic acid, either DNA or RNA, surrounded by a protein coat called a capsid, and some viruses have lipid bilayer called envelope. A virus can't replicate alone, instead it must infect cells and use components of the host cells to make copies of itself. What about viral structure? We have two types of viruses. First is called negative virus, which is composed of nucleic material and capsid, and the second is called enveloped virus, which is composed of nucleic material, capsid, and lipid bilayer called envelope. Both viruses have glycoprotein spikes that allow these viruses to penetrate host cells and cause infection. Are viruses dangerous? If so, why? When virus enters the cell, it stops all vital processes of the cell and uses most of the cell organelles in its unique replication process. In just 6 hours, it produces 100,000 progeny capable to invade other cells. When the virus exits the cell, it destroys the cell membrane during a process called budding. The virus is also able to make some mutation during the reassortment process, which allows it to evade and escape from the body's immune system and not be affected by some drugs. It has a high morbidity and mortality rate, so a virus is dangerous. But some viruses are useful because the gene of viral origin encodes information for a protein that plays a key role in long-term memory formation by moving information between cells in the nervous system. Herpes makes mice less susceptible to certain bacterial infection, including the bubonic plague, which is considered a common type of food poisoning. GB virus C is a pathogenic virus. Scientists also have found that GB virus C seems to make people infected with Ebola less likely to die. The virus is also used as a protective substance against the development of bacteria on food, such as bacteriophages. This process is highly dangerous because most foodborne viruses lack a viral envelope and thus are extremely stable in the environment. So, some foodborne viruses may be able to survive in foods, on hands, in feces, and on food contact surfaces and floors for long periods of time. And when they enter the host cell, they replicate. Norovirus and hepatitis A virus are the two most common causes of viral foodborne illness in the United States. How do viruses spread? First, infection via airborne droplets and particles. Some viruses can be transmitted through respiratory droplets that are released into the air when an infected person coughs or sneezes or talks. Other people can inhale these droplets, like influenza virus. Second, contact infection and smear infection. This occurs when an infected person touches or comes into close contact with another person, allowing the virus to transfer through skin or body fluids, like herpes simplex virus. Infection can also occur indirectly, however, as when an uninfected person touches a contaminated object, such as door handle. Any pathogens on this object could enter the host through wounds or mucous membranes. One form indirect contact infection is smear infection, which occurs fecal orally via the excreta of infected persons, like norovirus. Infection via contaminated water and food. This method occurs when viruses present in fecal matter enter the mouth, often due to inadequate hand washing after using the restroom or consuming contaminated water or food. They often occur in regions with poor hygiene conditions like hepatitis A virus and norovirus. Fourth, infection due to bloodborne transmission. Pathogens can also be transmitted via blood, tissue, or body secretions. This can happen directly through exposure to infected blood or body fluids like human immune deficiency virus, or indirectly through vector-borne transmission. Some viruses are spread by vectors such as mosquitoes or ticks that carry the virus from one host to another, like Zika virus. Now we will speak about two harmful viruses. First, human immune deficiency virus, which contains two copies of a single strand RNA genome within a conical capsid surrounded by a plasma membrane of host cell origin that contains viral envelope proteins. The RNA genome is 9,749 nucleotides long. The basic structural component of human immune deficiency virus. At first, it belongs to the family Retroviridi. and consists of two basic components: a core of ribonucleic acid RNA called the genome, and a protein component that surrounds the genome called a capsid. The genome carries the genetic material or information of the virus, while the capsid gives the virus its shape and protects the genome. The human immune deficiency virus genome consists of three major genes. First, group-specific antigen or capsid proteins. Second, polymerase gene proteins like reverse transcriptase proteases and integrase enzymes. Third, envelope glycoproteins. The capsid is made up of subunits called capsomeres. The second harmful virus is herpes simplex virus. Herpes virus have a unique four-layered structure. A lipid bilayer envelope derived from the host cell membrane. This envelope contains viral glycoprotein spikes and an icosahedral capsid made up of protein subunits called capsomere. The capsid protects the viral genetic material. This genetic material is a linear double-strand DNA genome. Between the capsid and the envelope is a protein layer called the tegument. This layer contains various proteins that are important for viral replication and the manipulation of the host's cellular machinery. Herpes simplex virus is the most common cause of erythema multiforme. The symptoms of erythema multiforme can vary, but the most recent understanding includes the following. The hallmark of erythema multiforme is the appearance of target or bullseye lesions, which have a central dark area surrounded by a lighter ring and an outer red halo. These lesions may appear as red or purple spots on the skin. In some cases, particularly in more severe forms like erythema multiforme major, the affected area may be itchy or painful. In more severe cases, mucous membranes such as the mouth, eyes, and genitals may also be affected, leading to symptoms like sores or ulceration. Some patients show systemic symptoms such as fever, fatigue, and malaise. How can you protect yourself from viruses? Frequently, you aren't defenseless against viruses. Your skin is a protective barrier that prevents viruses from entering your body. Your immune system also protects you by warding off invaders. Antibodies and vaccines likewise help your body better defend itself against certain viruses. This may include keeping a distance from others, wearing a mask, ensuring regular room ventilation, and coughing or sneezing into the crook of your arm. Hand washing is particularly essential, as is hand disinfection if there is any chance of contamination. في النهاية حابب أشكر الدكتورة سحر على مجهودها الكبير معايا، يعني هي ساعدتني بشكل كبير جدا في إنجاز الفيديو ده. وفي الآخر thank you.